All right, to start out, this is a video kind of just outlining how stuff in the gut can influence the brain and influence appetite and make you more or less satiated or more or less appetitive in terms of do you want to eat, do you not want to eat. And so typically this story starts within the gut. It could be in the brain. The brain could interpret signals and determine whether or not now is a good time to eat, or if now is a good time to kind of be satiated, to focus on other things. But for this example, this is about enteroendocrine cells, which are cells that are located mainly in the small intestine. It could be like in the duodenal region or the ileal region or the jejunal region of the small intestine. But this is where nutrients that are moving down the small intestine can influence cells that line the small intestine, where these are the enterocytes. These are the cells that actually make up the border of the small intestine, the epithelial cells. But these little red guys that I depicted are supposed to be enteroendocrine cells. So they're cells that are located like on the border of the small intestine. And this diagram definitely isn't anatomically very accurate. But these cells, what they do is they're positioned like there's some here, there's some here, there's some here, there's some here, where they're able to detect the presence of nutrients, the presence of chemicals at different spots within the gut. And then instead of releasing stuff like a lot of the out pockets of the gut release stuff into the gut, what they do is they detect the signal and they release a signal molecule into the space outside of the gut, which is usually a hormone. So if they're enteroendocrine cells, the endocrine part of that word kind of relates to the fact that they're acting as glands that release signal molecules into circulation. So they're releasing signal molecules into this little local environment, and some of those signal molecules could diffuse into circulation. But in this case, these little red guys are called L cells. So they're called L cells. And they're cells that are positioned in the small intestine, kind of lining the small intestine, and they're responsive to proteins, carbohydrates, and fats. And there's some kind of varying degrees in terms of like what they actually respond to, and it's largely fats and carbohydrates. But these cells, they detect fats, let's say, in the small intestine, and then they release GLP-1, so glucagon-like peptide 1, GLP-1, as a hormone in response to the presence of fats within the gut. And so GLP-1 is a hormone, so it's moving in circulation, but it has a really short half-life, meaning that it gets degraded really quickly. It's only around for about the half-life is about 2 minutes. So in 2 minutes, half of the concentration will remain, and then in another 2 minutes, only a fourth will remain, and so on. So there's these little enzymes that are positioned on the backside of these cells, these enterocytes, that actually break down GLP-1 really quickly. So GLP-1 is only around for a little bit in circulation, but it can have a direct effect on this nerve that goes down and innervates the gut. And this nerve is called the vagus nerve. And so the vagus nerve is really tied into the enteric nervous system, but there's sensory endings that sit right in here that have receptors for GLP-1. And those are typically G-protein receptors, G-protein linked receptors that are present on the actual dendritic regions of these sensory neurons that are part of the vagus nerve. And so the vagus nerve is composed of sensory neurons, visceral afferent sensory neurons. It's composed of motor neurons, composed of somatic motor neurons that control the laryngeal muscles and some muscles of the pharynx. But in this case, it's a visceral afferent sensory neuron. So it's relaying information from the gut into the brain. And so in this case, GLP-1 released from these enteroendocrine cells in the presence of fat is triggering this sensory neuron to send a signal up to the brain. And the cell body of this sensory neuron lies in the nodose ganglia. And that's largely because the sensory neurons that are part of this vagus nerve have their cell bodies outside of the what's termed like the central nervous system, because they're neural crest in origin. So that little cell body sits in a ganglia instead of a nucleus or a nuclei, because it's neural crest in origin. It's not neural tube in origin embryologically. But these visceral afferent sensory neurons relay that information regarding the presence of nutrients in the gut as a proxy for like the GLP-1 to this nucleus in the central nervous system called the nucleus tractus solitarius, which is this large area that relays a bunch of visceral information. It gets a lot of the sensory information from your body. It could be sensory information from your heart, it could be sensory information from your liver or from your gut in this case. But what it does is it integrates a lot of that sensory information, and then it sends that sensory information to other regions of the brain. And in this case, there's neurons that go from this nucleus tractus solitarius, and they go out and they're influenced by the vagus nerve, and they go out and they project to the arcuate nucleus, or they innervate the arcuate nucleus. And these neurons release a neuromodulator. So typically there's other neurons, like imagine one of these black guys reached out and it'll release something like glutamate onto these neurons. And then these green neurons are called POMC neurons. POMC neurons. And they're involved in decreasing food intake or increasing energy expenditure. So they're always on, they're always firing, but they increase the rate that they fire when you have a lot of food and you're no longer hungry. So they're kind of like a satiety, a set of neurons that are involved in satiety. So they're always on, but they increase the rate that they fire when you're full. And so this relay center where you're getting signals from the gut, I'm releasing GLP-1 here. That GLP-1 is having an effect on visceral afferent sensory neurons going up into the nucleus tractus solitarius, can have an effect because these neurons right here can also release GLP-1 as a neuromodulator in the brain. So the GLP-1 that's released right here is different from the GLP-1 that's released in this region from those enteroendocrine cells. It's the same molecule, but it's functionally in different spaces, and it's binding to receptors in both areas, G protein link receptors here and here, but the GLP-1 right here has a hard time influencing the GLP-1 up here directly because of the blood-brain barrier. Because GLP-1 is a protein, it's a peptide, it has a hard time crossing that blood-brain barrier. So this GLP-1 is functionally different from this one, even though they're the same molecule. But there's neurons that go from the nucleus tractus solitarius that are influenced by the vagus nerve that go up and release GLP-1 up here. And GLP-1 will increase the firing rate, so increase the firing rate of these POMC neurons, which will increase the satiety, or it will decrease food intake. So this is an example where the presence of nutrients in the gut can have an effect on your brain's ability to regulate whether or not it's a good time to consume food or a good time to be full and pursue other things. And it's part of the reason why some of those drugs that are GLP-1 agonists actually act, because their concentration is so much higher than normal. Some of the GLP-1 is able to cross the blood-brain barrier and get up here and act as a neuromodulator on these POMC neurons, these pro-opiomelanocortin neurons, and influence the rate that they fire, so the firing rate. And those neurons go over and they project to other regions of the brain, but they also project to this region called the periventricular nucleus. And what they do is they release alpha MSH, which is a neurotransmitter, which triggers these neurons to fire. And these neurons specifically are neurons that release oxytocin. So oxytocin is typically involved in bonding. It's involved in aspects of the birthing process. But functionally, in this sense, it's involved in consumatory behavior, so whether or not it's time to eat. And this alpha MSH will trigger these neurons to fire right here, which go from the periventricular nucleus over to the nucleus tractus solitarius. And they release oxytocin. So imagine all this purple stuff is oxytocin as a neuromodulator so not a neurotransmitter, but a neuromodulator that influences the sensitivity of these neurons in the nucleus tractus solitarius to vagal inputs. So it makes each one of these guys more sensitive to signals coming from the gut, which could further enhance the ability of GLP-1 to increase the firing right here, which can then loop around. So it's almost like a positive feedback loop in terms of the body's ability to sense what's happening in the gut and respond and influence satiety or whether or not you have increased appetite or decreased appetite. And there's a number of molecules that can cause a decrease in POMC firing rate. And those involve, some of those could be fructose, but there's definitely a gut influence on the brain's ability to be